Good afternoon. Sorry for not being able to be with you today, and thanks to the organizing committee for giving me the opportunity to present my work entitled Methane Producing Microbial Community for Marine Sediment as a Tool for Aquaculture Waste Treatment. Aquaculture and main salmon farming is one of Chile's economic resources, becoming the second largest producer of salmon in the world after Norway, with annual export valued at more than $6 billion. In addition, aquaculture is proposed as one of the solutions of the world food problem. But the industrialization of salmon farming has resulted in the generation of contaminants that can lead to eutrophication, reducing dissolved oxygen and increasing organic matter in marine sediment, which strongly affect the native biodiversity. For this reason, the need to treat the generated waste arises. The treatment of contaminated sediment is highly challenging, and for now they are usually untreated. An eastern alternative would be anaerobic digestion, with which the waste could be valued since biogas is generated that can be used as non-conventional renewable energy sources. But there is a problem, and it is that aquaculture waste has high salinity. Conventional anaerobic digestion is now be inhibited by increased salinity, with low methane production made due to the cell's plant molecule. On the other hand, methane emissions have been reported from the ocean sediment worldwide, especially in coastal areas. This suggests the presence of methanogenic archaea capable to withstanding high salinity, which could be used for the treatment of the waste for the aquaculture industry. With this background, the objective of our work has to determine the methanogenic potential of marine sediment impacted by aquaculture and to evaluate the effect of salt concentration on methanogenic activity and the structure of the microbial community. For this, treat sediment from under salmon farming cage from southern Chile and one sediment from an organic bass farm in the Bay of Marcilla were evaluated. As control, two sediments not impacted by the aquaculture industry coming from Valparaíso and Marcella were evaluated. We also included as a comparison an anaerobic sludge from the municipal waste treatment plant. Our sampler underwent a physicochemical characterization to determine conventional parameters. For each sediment slurry, methanogenic activity was determined in triplicate anaerobic bios operate in batch mode. 30 millimolar acetate has added as substrate. The methanogenic activity was evaluated at different salinity concentration between 0 and 35 grams per milliliter. During the incubation, biogas was measured periodically using a manometer and methane concentration was determined by gas chromatography. Methanogenic activity was calculated from the methane production kinetic a long time. Total urinate was extracted in the exponential phase of methane production. Bacteria, archaea, and methanogens were quantified by qPCR. The characterization of the community was performed by massive sequence of the before and before region of the 60S RNA genes. Let's move to the main results. Methanogenic activity could be detected in all samples except S4 and S5, even after more than 2,000 days of incubation. The activity was standardized by the number of total prokaryotes. Relatively stable acetoclastic activity was observed throughout the salinity range. but the latency time increased with increasing salinity. The quantification of the MCRA genes transcript evidenced the presence and activity of methanogen adapt to high salinity in all samples, except in the two samples where no methanogenic activity was detected. When comparing the initial sediment inoculum with the sediment sample at the end of the incubation, we observed an increase in transcript abundance in all marine samples, with the exception of the industrial sludge, where the decrease in the transcript was observed at high salinity. This confirmed the adaptation of the earth methanogen to salinity conditions. Finally, there was no correlation between the methanogenic activity and the abundance of the MCRA transcript. We already know that where is the methanogenic activity in marine sediment, but it is necessary to characterize the predominant methanogen who are the activity at high salinity. In X1, two methanogenic archaea were observed in the inoculum, but only methanogenium, a hydrogenotrophic archaea, was predominant at high salinity. In X2 and X6, there were no activity methanogenic archaea detectable in the inoculum, but after incubation, methanosarcina predominated, which can generate methane with all three payloads. Moreover, in X6, at the highest salinity, additional methanogenic genera emerged, shown as the methanogenium and a methylotrophic archaea that could not be affiliated to the genus level. In the case of the industrial sludge, the acetoclastic metanocyeta was predominant in the inoculum and maintained a low to the medium salinity. When salinity increased, we observed a strong yet swift methanosarcina and methanomethylophyllaceae can become predominant. The methanogens are only part of the active community that degrade the organic matter. Therefore, we analyze the structure of the active community globally. The sample clustered into the three groups in relation with the environmental variables. The fifth group corresponds to the sample with the highest methane production, coming from the organic farm in Marseille and the controls without aquaculture impact. In the second group, we find the sample under the salmon farm that produce methane and the industrial sludge. Under 30, the sample without methane production from under salmon cage that present specific microbial groups related to infections in fish. There were significant community differences between the origins of the sample except for S4 and X5. In all the samples, we observed a strong effect of salinity level on the active community structure. Here we show only the sample of S2. We select the O2s that present a significant change of the abundance according to salinity, and they are shown in this heat map. We observe that the samples were grouped first by the origin and second according to the gradient of salt concentration. From this heat map, it was possible to identify the potential metabolic function of taxa that were enriched with salinity. For example, we identified several allotolerant genera that can be produced of methane precursors, acetate and dihydrogen, by degrading different complex components such as amino acids, sugar, and peptides. This activity taxa might become partners of the methanogen from the treatment of the organic waste from aquaculture industry. To corroborate this hypothesis, an additional experiment was carried out using the inoculum that present methanogenic activity in order to treat a real aquaculture substrate. The recalcitrant substrate was the liquid fraction of S4 and S5 prepared at different COD level characteristics of the aquaculture industry. Methane was produced in all cases, but the X6 inoculum had significantly higher methanogenic activity with a tree substrate. On the other hand, the latency time was significantly high when using the industrial sludge as inoculum. X6 inoculum had the best compromise between activity and latency time. In conclusion, select marine sediments are sustained to treat the real aquaculture waste. Methanosarcina and methanogenic archaea appear to be resistant to high salinity. The environmental conditions will explain the difference of community according to their origin. Finally, our study confirmed the biotechnological interest of using the natural inoculum adapted to salinity to treat aquaculture residues through anaerobic digestion. We will be available to answer your question by email, or you can write to us on our WhatsApp in the real time right now. Thank you very much for your attention.